Mitosis is the process of cell division that makes possible the regeneration of body parts. This aim is achieved when the nuclear chromosome separates to form two identical daughter nuclei. This whole process is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase known as the cytokinesis occurs after a complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest, and this period is known as our interphase. In interphase, the genetic material of the cell increases. Then what happens in our prophase? During prophase, the chromatin condenses and becomes short and thick to form chromosome. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centroles, move apart to the opposite poles, forming a spindle between them. And that takes us to the metaphase. In metaphase, microtubules of the spindle interact with the chromosomes. This causes the chromosome to move in a line along the center of the cell, called the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of a protein structure called the kinetochore. The kinetochore holds the chromosome and attaches them to the spindle. Then we move to anaphase. During anaphase, the sister chromatids separate at the kinetochore, and the daughter chromosomes, along with the divided centromere, move toward the opposite poles as the microtubule shortens. The poles of the spindle also move apart, and this helps to separate the chromosomes. When chromosomes separate fully into two sets, one set at each pole, anaphase is then considered to be complete. And that takes us to telophase. During telophase, the daughter chromosomes arrive at the middle of the pole. Chromosomes decoil back into chromatin. Then the nuclei reform along the nucleoli. Then the nuclear membrane is reformed. This then takes us to the additional phase known as cytokinesis. Cytokinesis simply involves the partition of the cytoplasm. In animals, a pair of cleavage furrow formed in the plasma membrane divides the cytoplasm into two daughter cells, each cell containing a nucleus. In plants, new cell wall formation is triggered at the middle of the cell by the formation of a cell plate, which grows outward until it meets the existing walls, resulting in two daughter cells. Summary: mitosis is a process of cell division that makes possible the regeneration of body parts. P on the mat reminds us of all the phases in mitosis, as in prophase, metaphase, anaphase, and telophase. Interphase is the period of growth and rest, and cytokinesis occurs after a complete nuclear division. With this, we come to the end of mitotic cell division.